All right, according to my clock, it's 12 o'clock, so I'll go ahead and get started so we finish on time. It's my pleasure to be here today. I want to thank you for having me. I always like giving renal ground rounds to this group. Feel free to ask questions at any point. I'll also monitor the chat. So the title of my talk today is the first time I'll be presenting this research. The C-type natriuretic peptide receptor and endothelial sodium channel access in essential hypertension. So my lab has been studying this natriuretic peptide clearance receptor for the past 20 years, and we also are interested in epithelial sodium channels in the kidney. But I won't be talking about the epithelial sodium channels today. I'll be talking about the endothelial sodium channels. In this novel crosstalk that we recently discovered that led to the funding of the R1 grant application that we just concluded, I'll show you some data from that study. But I want to show the most important slide first, because it gives credit to the people that really did the work that I'm going to show today. I have a pretty big lab, and I'm very proud of everyone in my lab for their accomplishments and their contributions to various hypertension studies. So now if this laser will work, no, it won't. So I'll use my cursor. I want to acknowledge some, not everyone, but because of time, some of the main people that did the experiments that I'll show you the data today. Dr. Ling Yu in my lab, she did all of the electrophysiology, the PET studies I'll show. Nihar Kabbalah, she's a second-year doctoral student in my lab. Faisal Lucy, he's also a second-year doctoral student in my lab. I have a lot of clinical fellows in my lab, and I established an international collaboration with Argentine University in Turkey. So they send 1 to 2 fellows, clinical fellows that are in their last year of residency to my lab that are interested in learning how to become a physician scientist. So they want to learn basic science research. So they send them to my lab for 1 year. I train them, and then they go back and finish their residency and become a physician scientist. And I've had many of those over the last few years. Dr. Yunus Gawan, he's one of them. He came to my lab and did a lot of the work I'll show you today. He went back to his country, and now he's done with his residency. And he'll come back hopefully in January and do other great things. But a lot of my clinical trainees are well established now. They have went on to earn faculty positions at other universities, along with some of my doctoral students. Mohammed Golem is one of them. He recently graduated. Now he's faculty in Saudi Arabia. Ramesh Rafi, Dr. Rafi, is a current clinical fellow in my lab. So he did some of the studies I'll show you today. And then a lot of the undergrads, many of the undergrads in my lab have either gone to or completed medical school, pharmacy school, vet school, and very proud of them. They have contributed to various aspects of our hypertension studies. Current people in the lab that are doing some amazing work. Maggie was a summer student I had. She did great work that will lead to a publication soon. Salim, Hamza, Dr. Rasha Ali, she did a postdoc with me, and she did a fellowship in pediatric nephrology here, done a residency, and she's actually giving the grand rounds on December 19th at 8 a.m. So you get, if you're available, you might want to at least attend virtually and see some of the work that she did while she was in my lab. And I have many other people that contributed, but I can't take too much more time to spend on that. But I do want to acknowledge our collaborators, Dr. Nancy Denslow, and our external collaborators, Dr. Ladisa, Dr. Gertz, and Dr. Liang. We are actively collaborating on this particular project. And of course, I want to thank our funding. So one, our R1 from the NIH, NHLBI, and then the NIDDK. So the three objectives for my presentation, or four objectives for my presentation today, is one, to review alternative strategies independent of exclusive neprilysin inhibition, because that didn't go well. When we were interested in inhibiting just neprilysin by itself, and I'll mention some reasons why. But there are alternative measures that are currently being investigated where we have a neprilysin inhibitor and another inhibitor in combination to treat congestive heart failure and hypertension, and in some cases diabetic nephropathy. I'll have some data on that. The second objective is to identify novel mechanisms underlying the pathophysiology of essential hypertension and also salt-sensitive hypertension associated with the downregulation of the C-type natriuretic peptide receptor. So I want to convince you, hopefully by the end of my talk, that this receptor is not a biologically silent receptor that is only involved in the clearance of natriuretic peptides from circulation. In fact, it has a very much more important role in that signal transduction. I've spent the last 20 years studying that. The third objective is to recognize the significance of this novel crosstalk between MPRC and endothelial sodium channels. So we have discovered that MPRC, when it's bound to its ligand, activated by its ligand, natriuretic peptides, i.e. natriuretic peptides, leads to the inhibition of endothelial sodium channels. And these sodium channels are important in vessel stiffness, as you'll see from the talk today. And then the last objective is to evaluate the efficacy of using an agonist to MPRC to mitigate abnormally high endothelial sodium channel activity and stiffness in endothelium. So I have the QR code for your attendance. I also have it at the end just in case it doesn't work. For some reason it doesn't work sometimes, but I'll have it at the end just in case you missed it. But the organization of my talk today is divided into three parts. So the first part is to provide an overview of the role of MPRC in signal transduction. As I alluded to a few minutes ago, it's more than a clearance receptor for natriuretic peptides. And that's one of the main reasons we never saw a clinical trial for an MPRC inhibitor. Because as you'll see today, the inhibition of MPRC or the augmentation of MPRC can have beneficial roles. So it depends on the cell type and organ systems we're talking about. And then the third part of my talk is to convince you that this novel association, interaction, or feedback mechanism between the C-type natriuretic peptide receptor and endothelial sodium channels has important implications for blood pressure and in the context of regulating vessel stiffness. So I want to first show one slide on the natriuretic peptide family and their cognate, and talk about their cognate receptors. So these are the main natriuretic peptides: atrial natriuretic peptide, B-type natriuretic or brain natriuretic peptide because it was discovered in the brain, C-type natriuretic peptide, urodylatin, which is the kidney natriuretic peptide, and there's just one called dendroaspis natriuretic peptide, DNP, that's found in the venom of a venomous snake, the green mamba snake. They all share a similar homology. That's a cyclic ring structure of 17 amino acids linked by the sulfide bond between two cysteine residues. Now, what's important here is that when these natriuretic peptides bind to their cognate receptors, they result in the generation of important second messengers, and that is the important second messenger, cyclic GMP. So I want to point out here that there are three different subtypes or classes, or two classes, three subtypes of natriuretic peptide receptors. So the first is the GCA, or guanylyl cyclase coupled A, and then the guanylyl cyclase coupled B receptors. So these are the ones that mediate the effect of natriuretic peptide by generation of cyclic GMP. Now, the C-type natriuretic peptide receptor is not coupled to guanylyl cyclase, but it is coupled to the activation of phospholipase C and the inhibition of adenylyl cyclase. Now, I termed it an atypical G protein coupled receptor back in 2024. And I was working with Bob Lefkowitz, who won the Nobel Prize at Duke. And he was really interested in this receptor for a lot of reasons. One is that it doesn't have the 7 transmembrane spanning domains of typical G protein coupled receptor. It functions a homodimer. But there's 2 forms of it that we discovered. One is a 66 kilodalton form, and one is a 77 kilodalton form. So I'll talk about that later today in my talk. But for a long time, natriuretic peptides were thought to bind to MPRC, and MPRC were thought to remove them from circulation. That's true, but you have this neprilysin protein that does the same thing. Neprilysin degrades natriuretic peptides. So that's the pharmacological approach, is that if we can inhibit neprilysin, then we can increase the availability of natriuretic peptides. Now, in addition to having the privilege of meeting Dr. DeBold, we discovered ANF, atrial natriuretic factor, in 1980. I also have the privilege of working directly with Dr. David Bessler for a few years at the VA in Tampa, and he actually discovered 3 more natriuretic peptides are actually more potent and have a longer half-life than AMP. And those are vessel dilator, caliuretic natriuretic peptide, and long-acting natriuretic peptide. I won't talk about those 3 today because they go through different receptors. They actually go through nuclear receptors, not these cell surface guanylyl cyclase couple receptors and the MPRC receptor. So you can see here that natriuretic peptides are important in cardiovascular disease, and hypertension, many other places in the body. But one last thing I want to say here is that while CMP preferentially binds to GCB or NPRB, AMP and BMP preferentially bind to MPRA or guanylyl A. And all these natriuretic peptides bind to MPRC. Okay, so neprilysin inhibitor. This was initially the first approach, and it's not surprising that there was lots of enthusiasm of this approach to treat congestive heart failure and central hypertension, because if neprilysin does not act only on natriuretic peptides. So while it has beneficial roles in increasing the availability of vasodilators, it also increases the availability of vasoconstrictors like Ang2 and Endothelial 1. So it wasn't surprising that this clinical trials using condoxitril that showed a dose-dependent increase in circulating levels of AMP and increased natriuresis and increased cyclic GMP concentration also showed no relevant blood pressure change between placebo and hypertension patients. So the next approach was to give a combination of a neprilysin inhibitor and an ACE inhibitor. So this was a little more successful than neprilysin alone. And these drugs were termed vasopeptidases or vasopeptidase inhibitors. And the goal was to decrease systemic and renal vascular resistance, suppress aldosterone, and increase natriuresis and diuresis. So in preclinical studies, these compounds, they showed promise for heart failure and hypertension and renal disease, but they had side effects. And one of the main side effects was the angioedema. So the swelling of the lips, the face, upper respiratory tract, and sometimes the intestines. So we lost enthusiasm for these drugs. And drug companies then came up with a third approach. And this was to use neprilysin inhibitors in combination with the angiotensin 2 receptor blockers. So an ARB with neprilysin inhibitor. And this was a little more successful. So this strategy allowed us to switch patients that were on an ACE inhibitor and had associated angioedema, switch them over and to this ARB in combination with the nephrolysis inhibitor. And we found that the outcome of these clinical trials showed that there was a reduction in sudden cardiac death from progressive heart failure compared to the use of the ACE inhibitor alone, enalapril. So there is a new clinical trial, but they have not yet started recruiting, and that's for essential hypertension and type 2 diabetic nephropathy, as you can see here. So it's using sacubitril and valsartan, and it's interesting. It's going to be interesting to see how that goes. So we're talking about hypertension, secondary to diabetes here, or hypertension as a comorbidity of diabetic nephropathy. So an overview of MPRC. I just presented some important nuggets about MPRC, but these are some also important nuggets to consider that will be important for you to understand the rest of my talk. So MPRC binds to all natriuretic peptides. It's almost ubiquitously expressed. It functions as a homodimer, and it consists of a short 37 amino acid cytoplasmic tail. And that is the main part of the receptor in terms of mediating its role in signal transduction. So those amino acids contain peptides that are coupled to G-alpha I and G-alpha Q signal transduction. And as I alluded to earlier, there's two populations of MPRC. One is the 67 kilodalton form and one is the 77 kilodalton form. Okay, so back in 2001, Dr. Anand Srivastava in Canada identified several G-alpha I activating sequences within that 37 amino acid and cytoplasmic tail of MPRC. And these synthetic fragments shown here were found to inhibit adenylyl cyclase activity in heart particulate fractions and also inhibit vasoactive peptide-induced cell proliferation in rat A10 vascular muscle cells. So we also know that MPRC is coupled to G-alpha Q signal. So CAMP 4-23, that's a specific agonist of MPRC. We like to call it CANF. So I'll use those terms interchangeably. But this group shown that there's G-alpha Q sequences within the 37 amino acid tail of MPRC that plays a role in other pathways. The mitogen activated protein kinase pathway and AKT is the protein kinase B pathway. So I mentioned earlier that one reason that we never saw an MPRC inhibitor is because you would expect both beneficial and detrimental effects of inhibiting this receptor. So on one hand, while inhibiting the receptor would block internalization of natriuretic peptides and potentially allow for an increase in the availability of natriuretic peptides in systemic circulation, the blockade of this receptor will also attenuate its role in signal transduction. So here, CANF, the agonist of MPRC, is used to activate the receptor protein at the cell surface. And one group showed that MPRC mediated activation, or ligand binding mediated activation, reduces Ang2 induced nitrooxidative stress. And while another group showed that blockade of this receptor attenuated proteinuria and FSGS in an FSGS transgenic model. And they actually did a study where they used pyrimycin aminonucleoside, podocyte inhibitor or podocyte toxin in parallel with an FSGS transgenic animal. And what they found was that blockade of MPRC allowed for an increase in natriuretic peptides that mitigated or blunted the pure myosin amino peptidase toxicity or apoptosis of these podocytes, and also reduced proteinuria in these animals. So you can see here that MPRC is both good and bad if you block it. So we identified for the first time several important phosphorylation sites within the 37 amino acid tail of MPRC. And we know that MPRC is glycosylated, and that's one post-translational modification that regulates the stability of this protein at the membrane. But no one has ever investigated whether or not another type of post-translation modification, that being phosphorylation, regulates the function of this protein. So we identified 3 phospho residues that are indicated here in this slide, and also consensus sequences for protein kinase A, B, and G, along with CAM kinase 2. So what we did is we made, this was back in 2010, and since that time we made several mutant constructs and that were conjugated to flag a peptide, and in addition to a wild type construct, we made a mutant construct that was deficient in the tyrosine 521, deficient in phosphorylation at serine 521, and also deficient in phosphorylation at 533. So we use these constructs in experiments that I'll show later in my talk to investigate the functional regulation of this receptor by phosphorylation. So I made this grand rounds while I was on vacation in Turkey. So I'm going to sprinkle some pictures from my Turkey trip to wake you up. So on my summary slide, you'll see some pictures. So this is Zirke Castle. It was used in Turkey, and this was actually a watchtower during the Byzantine period. And it was also used by the Ottomans for accommodating tradesmen. Now you can go up there, you can touch things. This thing is, over 1,000 years old, right? So you can. It's not like a museum where you can't touch things and you can't walk around. You can actually go all the way up to the top, which we did, and you can explore. So it's a beautiful place. If you ever go to Turkey, I highly recommend that you go there. And this is a bridge on the way to the castle. So to summarize the first part of my talk, there are 2 functional populations of MPRC. MPRC is coupled to G alpha Q and G alpha I proteins. The 37 amino acid tail of MPRC has multiple phosphorylation sites that may be important in its function. And activation of MPRC by the agonist CANF was shown to reduce and to induce nitrooxidative stress and hyperproliferation of vascular smooth muscle cells. So the second part of my talk, I'll hopefully convince you that by the end of this these next few slides, that MPRC has an important role in the pathophysiology of aortic coarctation and persistent hypertension that develop in these children, even when they become adults. So one day I received an email, random email, and luckily I screened my emails because this was an important one, from Dr. John Ladisa from Medical College of Wisconsin. It was actually 5 years ago. So he said he submitted this grant application and the reviewers told him to add an expert on MPRC. So he said he read my dissertation. He had my dissertation, which I wrote on MPRC, on his desk, and he asked me if I would collaborate with him. So I did, and then we got the grant R01 funded, and we just exhausted it a few months ago. That led to several important publications and discoveries. What he did is that he found that he did RNA-seq analysis of coarctation tissue from children, and even after these children had the correction for coarctation by surgery, they developed persistent hypertension. So his RNA-seq data identified MPRC as being deficient. And And that's why he needed my help, because he didn't know what this receptor was and what it did. And he didn't have any tools to study it. So we collaborated, and I'll show you some of the stuff that was found. So coarctation, it's not that rare. It's actually pretty common, if you ask me. It's 5,000 to 8,000 births annually in the United States. It's a congenital cardiovascular defect that's characterized by constriction of the descending thoracic aorta. Treatment exists. So the best form of treatment is surgery. But even after correcting the coarctation by surgery, many of these patients develop persistent hypertension. So we thought that there's genetic factors that could be involved that could contribute to the persistent morbidity even after treatment. And there's genes that contribute to the pathophysiology of the aortic coarctation, although the etiology remains largely unknown. So Dr. Ladisa did an artistic analysis, and you can see here on the summary bar graphs on the right are normalized MPRC protein expression. So you can see the region with the high blood pressure, that's the proximal segment. It has significantly less MPRC protein expression compared to the distal segment where you have normal blood pressure. And he actually established a rabbit model that we use. So we have a rabbit model that recapitulates many of the clinical features that we see in patients. And you can see that here as well on the panel B, where compared to normal blood pressure, control rabbits, which is the bar on the right, the bar on the right here, coarctated rabbits with the high blood pressure have a significant decrease in MPRC protein expression, and even after correcting for the coarctation, MPRC protein expression remains low. So the model is that we use permanent suture or disposable sutures, and the permanent sutures represent persistent or untreated coarctation, and disposable sutures represent corrected coarctation. So this is it. We published this back in 2019. You can see here normal versus coarctation versus treated. And you can see mean arterial blood pressure on the right. You can see the coarctation group has 20 millimeter mercury increase in blood pressure compared to the control group. And although the blood pressure decreases after treatment, MPRC expression remains low, and then that blood pressure rises again, at least in people. We haven't had the rabbits live long enough, but the blood pressure is persistent in patients after treatment with coarctation. So this is the rabbit model. Yes.
Can you go back?
Yes.
Can I ask you a question?
Sure, absolutely.
It looks like you get some aneurysmal debilitation of the aorta after the coarctation that does not resolve. Can you
Precisely, yeah. So
is that
yeah, no, you're absolutely right. We found that, and we are following up with that in another study. I think he published a recent study. I'm not a part of that one. At the same time around this, maybe a follow-up paper from this one, and he noted that as well in that paper. So I think I have the reference at the end, or I can send it to you afterward.
so? I guess the question then is the difference in MPRC expression due to the aneurysmal dilatation. Is it due to the result stenotic lesion? How do you differentiate that?
Yeah, that's a good question, and the answer is we don't know yet. And we think it has to do with vessel stiffness. This novel interaction between endothelial sodium channels, deficient nitric oxide, dependent relaxation, and vessel stiffness, we think is part of the mechanism of this as well that we're seeing. And I think I have a couple slides later on to address this, and then a reference too for that. Yes.
How do you induce coarctation in rabbits?
So using sutures, disposable sutures or permanent sutures. So here's another slide.
Oh, go back.
Yeah.
We know, so you're showing the blood pressure gradient goes with it, you're not showing arterial blood pressure,
No, so we put
which is
telemetry,
which is very different.
very different, yeah,
So
so
we know with renal artery stenosis,
right,
if you resolve the stenosis after a prolonged period, if you wait too long before you have fixed hypertension. So did the blood pressure normalize?
so the blood pressure did normalize, but we're just looking at that segment, the proximal so you're talking about in that region or total body blood pressure?
Either, but you haven't shown, you said that blood pressure normalized, but
Oh,
the gradient
yeah,
normalizing doesn't mean that the.
no,
blood pressure-
so, yeah, so maybe I shouldn't say the blood pressure normalized. But the gradient was changed. We lost that 20 millimeter mercury gradient in that region, the proximal, compared to the distal region. Does that
So
answer your
it's not,
question?
well, but it makes it very different.
Right,
because-
right, yeah. Yeah, so we're just looking at that one segment. That one region.
Yeah, I have a question.
Yeah.
So did this study really try to see the correlation of NPR or C to blood pressure itself?
No, it did not.
So it is
No.
not intended, it did
No,
not
it was,
study.
that was not the purpose of it. We
Okay.
just wanted
So
to,
it does not say that
Identify-
despite the correction that the blood pressure remains high because of correlation to NPR.
not
It's
in this study,
okay.
not in this study, but in follow-up studies, we did address that. So here, this shows again that, I think it's a different paper, where, his group as well, they investigate systolic and mean blood pressure, elevations and coarctation, in corrected, rabbits, leading to, thickening of the vasculature and endothelial dysfunction in proximal versus distal. I guess this is the same question that you would have, right, David? So, yeah, it's just in that region. So, here we, administered the CMP, we looked at calcium mobilization, in particular calcium response to CMP, in these two photon microscopy experiments. And we see that there is a resolution in the calcium mobilization after administration of CMP in control versus coarctation in the proximal segment, but not in the distal segment. And we found there was increased calcium mobilization in the coarctation group compared to the control group. And we use this novel MPRC inhibitor here for some of these studies, along with the MPRC agonist CANF that I mentioned earlier. So to summarize this, their second part of my talk, RNA-seq data revealed a decrease in MPRC expression in aortic tissue from the proximal region of human coarctation patients. Rabbit model was established, and this recapitulates some of the anatomical features that we see in humans. And MPRC is downregulated in coarctation, and the decreased MPRC persists even after correcting for the coarctation. So this is Trabzon. It's one of the most visited places in Turkey. Beautiful scenic landscapes. And if you have time, I highly encourage that you visit that place if you're in Turkey as well. So the next part of my talk addresses an important crosstalk negative feedback regulation between the C-type nitric peptide receptor and endothelial sodium channels and essential, and I should say also salt-sensitive hypertension, as you'll see from some of the studies that I'll show you. So aortic stiffness and essential hypertension, there have been a few studies on this topic, and in 2022, Lundwall et al. Reported increased aortic stiffness correlates with a reduction in eGFR in hypertensive patients, and this study suggests an important role for aortic stiffness and the pathophysiology of hypertension-mediated renal dysfunction. Aortic stiffness is emerging as an important risk factor for organ damage and cardiovascular disease as well. So some groups have investigated or started to investigate mechanisms of hypertension due to aortic stiffening. Zoe et al. In 2017 investigated the role of rho kinase as a novel mediator of modulating aortic vascular muscle stiffness. And they had a follow-up paper around the same time that reported inhibition of the serum response factor, myocardial signaling pathway, and how that is associated with the reduction in aortic stiffness by targeting vascular smooth muscle cell stiffening and hypertension. And then another study, another group in 2014, who had all reported inflammation and mechanical stretch promote aortic stiffening and hypertension through a different pathway, P38 K pathway. Now we are using another method to in vitro method to mimic the coarctation rabbit studies and human studies that have been done, and that's to induce pronounced consistent cyclic strain in endothelial cells subject to a bioreactor. So we put these endothelial cells in a bioreactor, and that induces pronounced cyclic strain. And then we look at the activity of MPRC and now MPRC and ENaC. So it's well established that all studies have shown role of stiffness and endothelial cells lead to a decrease in the amount of nitric oxide that's released and available. And Showers et al. In 2019 showed hyperactivation of endothelial sodium channels lead to endothelial stiffening and impaired nitric oxide mediated arterial relaxation. Now, Murthy et al., a good friend of mine, Virginia Commonwealth University, he has been a pioneer in the field and has shown that MPRC is coupled to endothelial nitric oxide synthase activity that's required for the nitric oxide release. So you've seen this slide before because my lab is predominantly interested in the regulation of epithelial sodium channels. But now today I'm talking about endothelial sodium channels. And they are very different. So they do share some single channel characteristics. They're both sensitive to amiloride, so they're both inhibited by amiloride. They both are sensitive to dietary sodium. But the stoichiometry is different, the conductance is different, the proteases that cleave and activate them are different. There's many other differences. But here I'm showing you that ENaC, the epithelial sodium channel, can be regulated at multiple levels. One is N, the number of channels at the membrane. The other is PO, the open probability of that channel. And the product of N and PO is what gives us the activity, NPO. So here I show you that PIP2 is important in keeping the channel in open conformation. So if we deplete PIP2 levels, then we get less ENaC activity. And there's another protein that's involved here that we found was essential in serving as an adapter protein between ENaC and PIP2 because both are rare molecules. So ENaC is rare, PIP2 is rare, but there's this adapter protein called MARCs, actually a family of protein, MARCs-like protein and MARCs that allow for this interaction to occur. And I'll show you some data on that in these cells, these endothelial sodium channels today as well. So regulation of endothelial sodium channels. So in contrast, there being a feed-forward activation of endothelial sodium channels, epithelial sodium channels are inhibited by sodium, whereas endothelial sodium channels are activated by sodium. That's one difference. And another difference is endothelial sodium channels regulate cell stiffness and alter the synthesis and release of nitric oxide. And our lab have shown that both channels, type of channels, are inhibited by either disruption of the actin cytoskeleton and by various proteases. But the proteases present in the kidney and the lumen the urinary space, are different than the proteases that are present in the systemic circulation in these vessels. And then potassium, you can see here, the one group showed that submembrane cortex of endothelial cells, which is the location of nitric oxide synthase, softens with an increase in potassium. So we're looking at softening and stiffening of the endothelium, things that regulate it. Depolymerization of the actin cytoskeleton by cytoclasmin D. It causes softening of endothelial cells, where a low concentration of the serine protease trypsin causes an increase in the activity of these endothelial sodium channels and vessel stiffness. So our group has shown both, or shown endothelial sodium channels in both human aortic endothelial cells and human cardiac endothelial cells are inhibited by calcium mineralization and protein kinase C activity. So this study was done in rats where Sowers et al. Administered Western diet that had an excess fat and fructose for 16 weeks, and they maintained mice either on a standard chow diet or on a Western diet. And as you can see here, mice with an alpha ENaC, you can see here, mice, well-type mice that were maintained on a Western diet had an elevation in endothelium stiffness, and this was blunted in ENaC alpha knockout mice that were maintained on the Western diet. That's panel A. In panel B, we can see the nitric oxide dependent relaxation or acetylcholine is decreased in Western diet maintained alpha ENaC wild type mice compared to knockout mice. So our lab recently investigated whether or not activating MPRC by the specific agonist CANF could affect endothelial sodium channel activity. So for the first time, we performed single channel patch plant studies in primary cultures of human aortic endothelial cells from different normal healthy donors. And we found that ligand binding mediated activation of MPRC reduced endothelial sodium channel activity, as you can see here. And the conductance of these channels is bigger than the conductance of the epithelial sodium channels. So that's one difference that we can see from this IB curve. But they're still square channels. So ENaC is characteristic of being square, epithelial sodium channels and endothelial sodium channels, and they're both sensitive to familiarize. So here we measure ENaC activity in primary cultures of human cardiac endothelial cells. Again, after treating the cells with vehicle or CANF, and we found that CANF treatment decreased ENaC endothelial sodium channel activity, mainly at the level of N. That's wrong. Something got shifted here. I took out the number of patches, and that p-value is less than 0.05. So we did the reverse experiment. We knocked down, used siRNA mediated knockdown of MPRC to investigate the role of MPRC in serving as a negative feedback regulator of endothelial sodium channels. So here, cells were transfected with either non-targeting control siRNA or MPRC-specific siRNA, and then we did single channel patch clamp studies. And you can see here from the summary bar graphs, MPO activity was significantly increased when we knocked down MPRC by siRNA. So because MPRC internalizes when it's bound to its ligand, AMP, BMP, or CMP, we wanted to see whether or not ligand binding affects the internalization of MPRC in human cardiac endothelial cells. So we treated these cells with CANF or CANF plus the MPRC inhibitor. And as you can see here, there was a dramatic increase in MPRC internalization in cells treated with the agonist compared to cells treated with the agonist in combination with antagonist. So next, we wanted to use those constructs I alluded to earlier that we made. One was a wall type construct for the 37 amino acid domain of MPRC. Another one was the other three were phospho residues that were loss of function. So we mutated the serine 10 alanine, threonine 10 alanine, and the serine 521 10 alanine. And the take home message here is that blocking serine phosphorylation at serine 533 residue significantly decreased endothelial sodium channel activity after these cells were treated with the MPRC agonist CANF. So phosphorylation appears to have a role in the crosstalk between MPRC and endothelial sodium channels. So as I told you earlier, PEP2 plays a really important role in the regulation of epithelial sodium channels. So we wanted to investigate whether or not PEP2 also regulates endothelial sodium channels. So we depleted PEP2 concentrations by Wortmannin, using Wortmannin treatment compared to vehicle treatment. And then we measured sphingomyelin concentration and membrane fluidity. So depletion of phosphatoyl 4,5-bisphosphate concentration in the membrane resulted with Wortmannin resulted in a decrease in sphingomyelin content within the membrane, but an increase in membrane fluidity. So motivated by that data, we wanted to investigate whether or not the second messengers that result from the hydrolysis of PEP2 are increasing concentration in the membranes. So diacylglycerol is one of the second messengers that's increased after hydrolysis of PEP2 by phospholipase C. So we perform lipidomics. And you can see here, there are 12 different diacylglycerols that are all increased in availability after decreasing PEP2 synthesis or depleting PEP2 in the membranes. So we know that diacylglycerol activates protein kinase C, and protein kinase C negatively regulates epithelial sodium channel. So then we investigated whether or not protein kinase C activity was affected by the increase in diacylglycerol. Our hypothesis was that diacylglycerol increased phospholipase C activity. But I told you earlier that MPRC is coupled to the inhibition of adenyl cyclase and the activation of phospholipase C. So by giving the agonist of MPRC, we hypothesized that that would lead to activation of MPRC, activation of phospholipase C, hydrolysis of PEP2, so less PEP2, more of the production of diacylglycerol, and increase in protein kinase C activity. And that's exactly what this suggests. So PKC activity is increased when we give an agonist of MPRC. So now we wanted to mechanistically investigate whether Mark's protein is involved and how. So we mutated serine residues within the effector domain of Mark's. That's actually Mark-like protein. And we transfected these in mouse aortic endothelial cells. So compared to the wild type, there was an increase in endothelial sodium channel activity in cells transfected with a construct that had 3 substitutions of serine residues to alanine residues compared to wild type. And this is the model that summarizes what I just told you. So, increase in sphingomyelin enrichment led to a decrease in membrane fluidity. Depletion of lipid rafts, and the lipid raft lipid that we measured, PEP2, resulted in an increase in membrane fluidity. It also resulted in an increase in diacylglycerol, and that led to an increase in intracellular PKC activity and a dissociation of marks with the inner leaflet of plasma membrane and less endothelial sodium channel activity in the cells. So to summarize the last part of my talk, and we're going to finish on time, now a few minutes early, is that MPRC ligand binding attenuates the activity of endothelial sodium channels in both human aortic endothelial primary cultures, like human aortic endothelial cells, and human cardiac microvascular endothelial cells. So siRNA-mediated knockdown of MPRC increased the activity of endothelial sodium channels in human aortic endothelial cells. Loss of phosphorylation at serine residues 533 of the cytoplasmic tail of MPRC resulted in an inhibition of endothelial sodium channel activity in human aortic endothelial cells. PEP2 depletion using wortmannin resulted in an increased level of bioactive diacylglycerol, and that in turn activates PKC. So we saw an increase in PKC with no surprise, and this was done in aortic endothelial cell membrane fractions. And then the depletion of PEP2 by wortmannin treatment resulted in a decrease in sphingomyelin content and increase in membrane fluidity in these mouse aortic endothelial cells. And overexpressing a construct that mimicked constitutive phosphorylation of Marx-like protein 1 resulted in a decrease in endothelial sodium channel activity. I can't say ENaC because, we're talking about two different channels here, mainly at the level of N in these mouse aortic endothelial cells. So future direction, we have a lot of them. So we just established, so typically we work with transgenic mice, but in collaboration with Dr. Aaron Gertz and Dr. Mingyu Liang, we actually established a rat model, MPRC overexpression rat model that we're using for the first time to corroborate this novel feedback inhibition between MPRC and endothelial sodium channels. In addition to that, we want to investigate ligand binding of MPRC kinetics and the recycling insertion of the protein at the membrane. We want to investigate endogenous endothelial sodium channel activity in different models, and not just different models, but different models maintained on high sodium, low potassium diet. And we want to investigate the efficacy of using anti-hypertensive drugs or repurposing anti-hypertensive drugs to normalize endothelial sodium channel and MPRC function and patients with aortic coarctation. And we finally want to evaluate the efficacy of repurposing anti-diabetic drugs like dapagliflozin, metformin, and normalizing endothelial sodium channel activity and MPRC activity, and also blood pressure in salt-included hypertensive diabetic animals. So we have found that in the kidney of multiple models of diabetic nephropathy that are salt sensitive, including DVD mice, that when we salt load these animals, they develop profound hypertension after just 10 days. And if we give metformin or dapagliflozin, we normalize the ENaC activity and the epithelial sodium channel activity and normalize the blood pressure in those mice. So these studies are all published, but we don't know how endothelial sodium channels and vessels or in cells of microvascular resistant cell arteries are affected. So that's another future correction. What that